Thank you for attending this morning. We're going to talk about CRISPR and try to put it in some perspective. I've sent you several articles. One of them is a stat summary of a discussion of four of the principals who were involved in CRISPR or who are involved in a very deep way. The first two are the discoverers of CRISPR, which is a bacterial defense mechanism. So both Jennifer Dunda and Emmanuel Charpentier were working on a very fundamental question, which is how do bacteria protect themselves from infection by viruses, something that we're all interested in, how we protect ourselves from viruses. And they discovered a pathway whereby the invading organism is inactivated because the bacteria previously seen it and used little bits of RNA to chop up the DNA of the invading organism. But the way they do it led them, and then later some others, particularly Feng Zhang and George Church, the other two people who were referred to in the article, to think about how you could edit the genes of human cells. Now, let's take a step backwards and say, what did people really want to do? Well, for a long time, if you think about the revolutions that occurred in biology in my lifetime, the first was the discovery of DNA goes to RNA, goes to protein, goes to phenotype. After about 25 years, it was found in the mid-50s, really by the beginning of the 80s, people were beginning to use it to produce proteins, and today that's half the pharmaceutical market. Over some drugs sell up to 50, I would say combined $150 billion worth of sales from that single discovery. That took about 25 years. DNA goes to RNA, goes to protein. It was about 15 years from the discovery of reverse transcriptase, turning RNA into DNA, and then being able to insert that into bacteria and produce purified proteins, purified antibodies. And that's the backbone of much of modern pharmaceutical work today. It is also has been a tremendous driver for understanding the processes of biology of all living organisms from plants. But one of the hopes early on had been that you could replace genes that were causing inherited disease. The revolution of the DNA revolution was able to pinpoint, especially with hemoglobins, changes in the DNA sequence, which led to the change in the protein, which led to the sickle cell disease, or sometimes hemoglobins wouldn't be made, led to a disease called thalassemias, particularly a particular variant called beta thalassemia. And the hope was that once we discovered the whole DNA, we discovered retroviruses that could insert themselves into DNA, or other ways to make the protein, that you could permanently change the genetics of an individual so that they were able to overcome their inherited disease. Now, there's a very, very long list of inherited diseases, and that list is getting even longer as we understand the processes of disease. We've sequenced the genome. There's genome association, so that the list of diseases is getting longer and longer. But the hope for gene therapy, with very, very few exceptions, has not turned out to be the case. And one reason for that is the way that gene therapy works is using a virus vector to put in a whole gene, not change a gene that's there, but put in a whole gene and put it in permanently. And the problem is that really specific. Genes have gone into the wrong place for a disease called combined immune deficiency. The number of the problems were cured, but some of the genes went into the wrong place, and certain children who received that gene therapy later got leukemias, and we really know now that one of the ways leukemias are formed is putting an enhancer and promoter in the wrong place, turning on a gene that causes cells to grow apparently. So that had not worked very well. So although the DNA revolution was quite amazing, really changed a lot of things. And then the second revolution was the use of DNA to make proteins and antibodies. It's really changed lives all over the world. In about 11 years ago, the Dundas and Charpentier discovered CRISPR, and Feng Zhang proposed to use it for human gene editing. That was 11 years ago. Just recently, a week or two ago, the British approved a drug produced by a company that Charpentier started, but it was actually delivered by another company called Vertex to treat one very specific idea. And Kim, who is on the phone, has written about that. We wrote about the use of CRISPR for the treatment of two different inherited diseases. One of them is the very famous sickle cell disease. And sickle cell was one of the very first diseases that we really pinpointed accurately with protein and DNA chemistry. A single change in a single nucleic acid changes the structure of the protein of globin, which is in hemoglobin and red blood cells, and causes it to form crystals with others of its kind, and those deform the red blood cell and can lead to clots and a series of very serious disease. If you're homozygous for that, it'd be very dangerous. And it's quite prevalent in Africa because if you only have half of that, you have about half of the disease, and you're more resistant to malaria. And it's become quite prevalent, and it's present here in the US. I don't know the exact numbers, but it's been something we've tried to treat for a very long time. The prior successful gene substitutions were really draconian. What they would do is they would really do a bone marrow transplant. You would basically kill somebody by wiping out their bone marrow precursor cells or hematopoietic stem cells with a drug like busulfan. Then you would take cells that from a different person that had the intact gene, put that in, that would repopulate the blood cell. And that's a very risky procedure. It was originally created many years ago when I was a young professor at Dana-Farber. And I would say there was usually about a 5% mortality. It's down to less than 1%, but it's still pretty dangerous, the preconditioning. So it was very rarely used. But it did work. There are a few drugs that can help somewhat, not really particularly. But what CRISPR allows you to do is do almost the same thing. Take the cells, the cell precursors, the hematopoietic stem cells from an individual with sickle cell, take them out of the body, change them. And what CRISPR does is it's very specific. You can either put in whole genes or you can put in a single change. You can delete whole genes. There's a whole series of things you can do with the CRISPR. And there's a whole industry that's now involved. And I would say there are a number of people using this bacterial system. It turns out that many bacteria have systems like this. Our CRISPR-Cas9, which was the original one that was found, has a guide RNA that locks you into place and a place that cuts. And people have figured out how to change 1 nucleotide, 5 nucleotides, a whole gene, to add it, to subtract it. So what they did is they wrap the CRISPR in a vector, they insert it into the stem cell. They then proliferate those stem cells. But the patient who has sickle cell has to be treated with a drug called busulfan, which prepares them to receive the healthy or the repaired hematopoietic stem cell. That is equivalent to a bone marrow transplant, or very, very similar. And that's true of a lot of the technologies I'm going to talk about. You have to prepare the person by treating them and knocking out their hematopoietic stem cell capability and replacing that, or partially replacing that, with the modified cells. That then allows that hematopoietic stem cell to new ones would be changed. And in particular, what they did, this particular one, activated a type of globin molecule that's present during fetal life, which gets turned off as adults. But if you turn it on by changing the promoter enhancer region, by changing the nucleotides with CRISPR, you can have that bone marrow stem cell, and therefore all of the replaced cells produce healthy globin and basically change the disease. Now, it turns out that that one change, and it's a drug called Casavy, produced by Vertex, has been approved in the UK for 2 diseases. You need to make healthy globin that's no longer the sickle cell, or if you're not making a globin molecule altogether, it allows you to make a fetal globin. So you treat a disease called beta thalassemia, or you treat sickle cell with exactly the same drug. That has taken about 11 years. Now, in the interview that I've sent you, both Charpentier and Dunda said, we just didn't expect it to happen so fast. Well, what I remember is from the time we could clone DNA and insert it, it was about 11 years before the biotechnology revolution took place. When we made hepatitis B antigen, we made erythropoietin, we made growth hormone, we made all sorts of drugs, and then later it's turned into making antibodies. So it's about the same time. It's not really that fast. It does take time, but it's revolutionized medicine. But it's also, just as the DNA revolution did, it revolutionized science altogether. I've been reading these amazing papers where people are now understanding very, very complicated biological processes by knocking out, using CRISPR, which is knocking out entire families of genes and looking at what happens. I was just looking at a paper over the weekend where people used almost 500 different kind of CRISPRs to look at a kind of an RNA and knock them all out, and then figured which one of those 500 is the one that's most important. So you can do amazing things in biology, and it's really changed biology, not just in animal cells, but I have to say in agriculture. The biggest impact on human life in the near term for this technology will be agriculture. And for one very important reason, and that is, as you all know, there's been a big controversy on genetically modified organisms. By putting a gene in for plant resistance to a pest or a pesticide, changing its growth characteristics, has been anathema to countries that are agriculturally based, like most European countries, France and Germany, Great Britain, but also Japan and some other countries. And that's prevented places like Africa from using the technology that would like to export their food for income to those countries. And so it's really the European laws and other laws against GMOs have been really frightful for their impact in poor countries. However, using CRISPR, it's a little bit different. CRISPR goes in, it doesn't stay there. It changes the sequence and then it's gone. That's very much like radiation. So the European authorities have said, well, since you're not leaving anything there that couldn't have been there before, it couldn't have happened by chance or couldn't happen by radiation, which we allow, we will allow you to do that with CRISPR. And I have friends that have created companies, and there's several companies around the world now, and it's sort of a silent revolution in agriculture using CRISPR to change the genes for either better productivity, better nutrition, or resistance to pests or pesticides. It's a booming industry. And that's a very interesting business, because what you do is you sell the seed. You don't necessarily sell the food. And you're not going to see the label on the CRISPR modified food that you're eating that says this is GMO. It'll say it's non-GMO, because it's not what's called GMO. It is altered, but it's no different from irradiating. We'll see that for livestock as well. We'll be able to improve livestock and production, and many of the things that GMO could do, this can do, and can do with approval. So CRISPR is changing much of life. Now, what are the downsides of CRISPR for human beings? Well, it's obvious that it's not without cost to treat a human being with busulfan. There is some mortality involved in that, whether it's 0.1% or 1%. Some people will not make it. Now, is it worth their lifelong suffering from crippling sickle cell disease? It might be. Are there other treatments that could use that aren't so drastic? Maybe. But having to prepare somebody with busulfan so you can insert their hematopoietic stem cells is difficult. More recently, some other downsides have appeared. That very process of taking the cells out, causing them to proliferate in culture, has activated some viruses. In particular, in some cases, has activated herpes virus 6. Now, almost none of you have heard about herpes virus 6. You know about herpes simplex, you know about herpes zoster, but you don't know about herpes virus 6. Almost all of you have it. So if any of you go through this process, some of you are going to have it activated. And it turns out it's very serious neurological complications once it's activated, and it can affect the brain and it can kill you. So that is a definitely negative side effect. There's another side effect, which I would point out, and that is herpes virus 8 causes cancer, causes Kaposi sarcoma. And for a couple of reasons. First of all, if you're immune suppressed with HIV, you aren't suppressing your herpes virus 8, but the activation that I discovered, the TAT activation of HIV, will activate herpes viruses and vice versa. So there's something called transactivation, which is mutual. So if there's an underlying population with herpes virus 8, now why am I perseverating about herpes virus 8? Very low percentage of Americans, it's a sexually transmitted disease, so the same group of people who can get HIV can have Kaposi sarcoma. And years ago, when we were just looking at HIV to begin with, a lot of Americans who got HIV or HIV positive also had Kaposi's. And that's the result of what I was mentioning. However, in Africa, about 50% of people in sub-Saharan Africa are positive for herpes virus 8. So exactly where you need this sickle cell treatment is a problem if you have to do immune suppression. There's another big problem that many people talk about, and in the interview, George Church talks about it a lot, which is the cost. Think of what it means. You take a patient, you treat them to ablate their bone marrow stem cells. They are then susceptible to lots and lots of infections. They have to be very carefully isolated for some period of time. It used to be a couple of months, now it's down to a week or two. But still, you've got to be very careful that they don't get another infection while you've knocked out their immune system. Okay? Then you've got to take the cells, grow them up, modify them, and put them back in, and then wait for them to take over so the person becomes immunocompetent again. That process not only is dangerous, it's expensive. These treatments probably in the US will cost between $1 million, some people think $2 million a person. How many people can afford that? Almost nobody. So can insurance afford it? Well, you know, if you're a pharmaceutical company, you can say, well, to treat this person would cost a lot of money, therefore it's worth investing some money. And that's a typical argument for high cost drugs today. For many drugs are costing hundreds of thousands of dollars today that do genetic manipulation of one kind or another. But that is not something for most people with sickle cell. In fact, it's a great limitation. So it's difficult, it's expensive, and it's dangerous. Are there ways around that? Well, there might be. We're now at the point where people are beginning to think, why do I have to take the cells out? If I can target the vector that carries CRISPR and then edit directly to the cells I'm interested in, maybe I can just give a simple injection And I don't have to go through that whole process. Now, what's the problem with that? Well, that can work for some things. For example, it can make some proteins. For example, if you have the ability to put what CRISPR allows you to do is put in genes, which can make a whole protein, you can begin to have cells that you've injected make that protein. And there are a couple of examples of that, which I'm going to describe. One of them is something that we've written about, which is a treatment for cardiac fibrosis. Now, it's not approved for humans, but it's been done in mice, and it's pretty interesting. What they've done is they've made a liposome, which is how you package the messenger RNAs. And now what we're talking about is something pretty new. And this is one of the more interesting things about science, is that you can take advances from different fields, combine them, and getting something brand new. What are the elements? The first element is a liposome. They've been around for a long time. I remember starting a company using liposomes with one of the, Bob Langer, a MIT professor who's an expert at that, maybe 30, 40 years ago. But people have learned a lot about liposomes. But you can take a liposome, it's sort of like a little lipid vesicle. It protects what's ever inside, made out of fats. And what you can do is you can add an antibody to that. So it will bind certain kinds of cells. That's how you can target it. So targeted liposome or targeted other therapies. Now there's going to be a revolution where people are figuring out how to target within your body. So you don't have to take the cells out. You can just take the CRISPR and it's going to change the sequence or put a new protein or a new gene in there, and then target it to the cell that you want, whether it's a neuron, whether it's a muscle cell, whether it's a liver cell. It turns out liver cells are the first ones people use, and they use it for different reasons. Second is RNA technologies. We are all familiar with the use of an RNA to make a protein, and that's the vaccine. It can make a protein for a day, or can make it, if it's a self-amplifying RNA, can maybe make it for 20 days or even longer. Or if you combine it with CRISPR-Cas9, you may be able to make a whole, put the gene in and keep it there permanently. So that's a second revolution. The liposome, the CRISPR-Cas9, the mRNA technologies are all now being combined so we can be able to change the proteins and processes of the human body targeted. Now that's a little bit pie in the sky. It's only been done in 1 or 2 cases, but I've talked about one in a mouse where you put in, with an injection into the mouse, a liposome, which is targeted pretty specifically. It makes an antibody, and that antibody attacks the proteins that cause fibrosis after a heart attack. So let's take that one step at a time. When you have a heart attack, you damage the tissue. In humans, especially, not some earlier animals, if you don't regenerate muscle tissue, you regenerate a repair, like a scar, and it doesn't really function the same way. If you ever look at a heart that has had a heart attack and it's beating, you'll see a bubble. Every time the heart beats, there's a bubble, and you're terrified that maybe that bubble will break, and sometimes it does. So you get a bubble where it's not really muscle in the beating heart. Not a good idea. The idea is you put in a cell which makes an antibody which targets the protein that stimulates the scar formation. And that has worked in mice. Will that work in humans? People are going to try it. Okay, that's the first one. The second one is even a little bit more interesting, and I'll get to that in a minute. But the whole idea is, can we take the power of CRISPR-Cas9 plus liposomes plus in vivo targeting and target it to the right cell? That is what science is trying to do today. We're not there yet. We're just beginning. We have little hints that it will work. But if we can do that, what can we do? Think of what the difference would be between a shot which solved sickle cell, and the process that now solves sickle cell. The difference between $1 and $2 million and perhaps $10 and $20 in the actual process. Because why do things cost so much for the patient, CRISPR? It's because you've got to have the medical system to take care of the person who's immunosuppressed for a while. You've got to have experts who know how to prepare the cells. And then you've got to take care of it. It's a whole medical system. It's a very expensive system. It's very people intensive. And it's available only in a few places in the world and for a few people. That isn't the case if we can target these genes specifically. Now, are there other cases where we can target genes? The answer is yes. There's a company called Alnylam and other companies now that makes what's called silencing RNA. It's a little bit of RNA that sticks to a messenger RNA and it causes it to fall apart. Well, you can turn off genes by doing that. And if there's a gene that's causing a problem, you can do it. Now, people have had great success in doing that with liver. Why liver? Well, it turns out liver, because of its function, has what's called fenestrations. If you speak French, it means window. Fenestra, it's a window and allows things to go in easily. Also, it has a receptor for something called GalNAc, and it's a sugar. You can put a sugar onto the RNA. And it's turned out there are maybe 4 or 5 drugs that have been developed by using a liposome that goes into the liver fenestration, that delivers a silencing RNA, and that turns off the gene that the liver is making. And now people are using it to make a protein in the liver. So the liver produces a lot of blood proteins. A lot of the proteins in your body are made by liver. And they're actually wrapping it, putting a messenger RNA or a piece of DNA in there, usually messenger RNA, that allows it to be made. And so you can begin to think about using single shots. People are now using the same strategy with silencing RNAs to treat things like glioma or other brain diseases by targeting the certain brain cells. So there is a reality there. There's 5 or 6 drugs that have been approved. And there are other companies. Usually what happens, if one company does it, another company does it, you think that the patents would stop that. They don't really. Patents are really pretty specific. So you can, a lot of ways to work around patents. So that's a way of targeting liposomes to deliver RNA to liver in particular, but now more and more tissues. And I would say in the next 10 to 15 years, we're going to see a tremendous advance in that technology. Now, where can that lead us? That can lead us to something that I'm very excited about, which is replacing protein drugs with RNA drugs. Why do I say that? Well, think about what a vaccine is. A vaccine is just a protein that you may have the body make itself. So rather than putting in a dead virus or a piece of a dead virus, you put in the RNA, your body makes the protein, just like the virus would make the protein from the viral RNA. It's a piece of RNA that makes a protein, your body sees it as foreign. Can we use that RNA to make therapeutic proteins and antibodies? And I believe the answer is yes. That we will, in the future, be able, with a single shot, to make a protein, which is now the subject of half of the pharmaceutical industry. And soon to be more than that, it's antibodies and proteins, things like growth hormone, insulin, you name it. Why can't we just use the RNA to do it? And what's the advantage of that? Well, I have made antibody drugs. It cost me about $200 million to build a factory. You've got to make, it's very difficult. You have to make the protein, and proteins have to be, and believe me, when you look at the FDA regulations for making any drug, it's a nightmare. You have to know where the water is from, where the salt is from. Everything has to be controlled. And if you're going to stick it into a human body, you're happy they're doing all that. But when it comes to making a bunch of proteins, it's expensive. It's not cheap at all, whether they're antibodies or protein drugs. However, RNA is a chemical synthesis. In a room the size of the garage, you can make 1 billion doses. And then with a little bit more, you can wrap it up. You need a sort of another facility that costs maybe $10 to $30 million, depending on where you are to package it all. But eventually you can take treatments that are costing hundreds of thousands of dollars, or even $10,000, $20,000, and get it down to $10 or $20 if you have an RNA delivered injectable drug. And that's where the pharmaceutical industry should also be going, not necessarily in the US, not necessarily in Europe, but certainly in China, India, Indonesia, South America, a lot of places that simply can't afford the best medicines. That's what the countries should be doing. And are they doing it? Sort of. They don't really see the vision yet, and you don't have the right kind of support. But I'm confident in the next 10, 20 years, in your lifetime, you will see some of these treatments that are now available only for those who are very expensive. And in India, they've actually got it down to as low as you can get it, companies like Biocon, but they're still very expensive. They're still expensive to produce. If you can lower the cost by a factor of 100 or more, you can treat most people, whether they have diabetes or many, many of the other ills, rheumatoid arthritis, inflammatory bowel disease, etc. Now, finally, we're coming to one other area. We're coming to the area which I've just written a book about. It's the CAR-T book. I meant to have a copy here. I don't have it behind me. I don't know where I'm talking. But we've been working very hard on CAR-T, and Kim Hazel has been working with me on a bunch of, not Kim Hazel, she was working on the eye. It's Amara Muta has been working on the CAR-T book, and we've written a whole lot about it. And what are CAR-T cells? Well, let me take a little diversion, and it's not different from what I'm talking about. It's actually using the same series of technologies. So what is CAR-T? Very early on, people recognized that, let's say in the early 1900s, 1905, Ehrlich and others, who were actually students of Koch, one of the great bacteriologists, got a Nobel Prize for understanding some aspects of the immune system, and said, why can't the immune system fight a cancer cell? Because a cancer cell is obviously a little bit different from your own cell. And in fact, it is true. So it's been a hope for many, many years that we would be able to use the immune system to fight cancer. In fact, the Dana-Farber Cancer Center, where I worked in the 70s, 80s, and 90s, was devoted to that. And their idea was to take antibodies, put a poison on them, and use those to attack specific cells. It took a long, long time, maybe 30 years, to find the right combination of antibodies and poisons, but it's now beginning to work. That use of an antibody immune system to kill cancer cells is working. That isn't CAR-T. That's just a whole different kind of cancer therapy that's added another arrow to the quiver in fighting cancer. However, at NIH, Steve Rosenberg thought maybe T cells that have a natural killer function could be used. So he took the T cells out of the body, educated them, proliferated them, put them back into the body. And it worked a little bit, but it had a tremendous side effect. It was pretty nonspecific. People got all sorts of cytokine storms. And I had a friend who had got many different kinds of cancer therapies, and he said the worst side effects he ever had was with what was called TIL cell therapy. Well, that has now evolved. And what people have learned is that T cells to attack and kill need two signals. They have to be presented by an antigen, and then they have to recognize the foreign substance. And people thought, well, maybe I could combine that. Maybe I can modify a T cell so it triggers the killing, and it can see with the antibody portion what's different about the cancer cell. So they've built these cells called CAR T cells. They're T cells, CD8. But in order to make those work, they've got to insert the gene. They've got to do gene therapy on the T cell. And we've just talked about what gene therapy can be now. CRISPR-Cas9 is really great for putting genes in. People were using retroviruses, they were using all sorts of other things. Now they're using CRISPR-Cas9 to put in the genes they want very efficiently. That's the connection. the first connection. So they're taking the T cells out of the body, modifying them, and putting them back in the body. Well, that's expensive, as we just gone through. That is dangerous, as we've just gone through, but it does work for refractory cancers. And in particular, there's 6 drugs now approved, maybe a few more, but now 6 for various kinds of B-cell cancers. So that has been a pretty effective therapy. Use CRISPR-Cas9 to change the T cell to put the right antigen on. Now, there are a lot of problems with that. First of all, the problems are the ones we discussed. You have to prepare the person, you're going to put the T cells in, it's dangerous, but if you're going to die of cancer, it may be worth taking the risk. And if you have the money and the healthcare system is working, maybe that will work. So it does work and has saved a number of lives. And it is especially used for people who failed other more traditional types of cancer therapy, sort of salvage therapies. But there are a number, and there are a growing number, and there's a huge number that are being tried. And that is where CRISPR-Cas9 is mostly used today in medicine, to change the T cell that's going to fight the cancer. And in this list, I've made a list here, which I will just read off. Okay? I think I have it here. It's been used for breast cancer, liver cancer, esophageal GI cancer, prostate cancer, bladder cancer, a bunch of different kinds of leukemia, B cells and T cells, lung cancer, kidney cancer, and some brain cancers. So it's been used for a lot, and there's a tremendous number of companies doing this in treatment. Will we get to the point where we can modify those CD8 cells with a single injection? I think the answer is yes. I think we'll be able to do that. And I think that the next generation of cancer therapies will be single injections that modify the T cells. Now, there is another problem. When you modify it, the great thing, the reason it's really different from gene therapy is it's not permanent. It doesn't have to be permanent. You can turn it on and off. And one of the things that Amara has written a lot about is advances to turn the T cells on and off. One side effect of CAR T cells is the cells are programmed to kill, and they may kill things you don't want them to kill. So you've got to be able to turn them on and off. And people are now working on all the control systems, decades of research in gene regulation and how drugs can be used them to turn them on and off work well. Another limitation, cancer cells can, if you attack them, can mutate. So the target you're using gets killed, but the cells, some of the cells can change and grow even though they don't have that particular target on the surface. So people are now working out how you can switch and use multiple targets, or switch targets, so you can switch them on and off. You can use multiple targets. Some very elegant ways which we describe in our book, CAR T cell cancer therapy. And we're talking about how you can use it now. People have also noticed you can target normal cells in people where their immune systems are acting up with autoimmune diseases. Can you kill those cells that are causing autoimmunity? For rheumatoid arthritis, for MS, for irritable bowel syndrome, for a lot of different diseases? And the answer is yes, you can do that. And animal systems, and in some people, they're beginning to try that. You can use the same kind of killing. There's another thought that's occurred to people. Gee, CD8 cells aren't the only cells that kill. There's gamma delta T cells that kill. There are natural killer cells that kill. Macrophages gobble up things that they see. There's a whole series of killers in your body to help protect you if things go wrong, either from cancers are caused or invading organisms. And so you're beginning to adapt many of these things. And we talk a little bit about that in this book, the CAR T book. It's available on Amazon and other places. So that's another use for CAR T. So just sort of conclude my thoughts on this. This is a real revolution in biology. It's as fundamental, and actually, if you actually think about the revolutions that have occurred, DNA goes to RNA, goes to protein. The understanding of inheritance has opened up the field of biology, all living systems, and has led to phenomenal increases in the number of drugs and the kind of drugs we have and diseases we can treat. There has been the gene delivery technology, the liposomes, and understanding silencing RNAs beginning to have an impact in medicine. You can remember very well the blinding insights that have come from using RNA as a drug. It turns out it's useful for vaccines, especially for some aspects of vaccines. Not perfect yet, but moving along, and you can actually use it, and it's been used in billions of people in our lifetime in a very short period of time. What people aren't aware of is there was about a 10-year prologue where the vaccines only worked for a short time, and therefore pharmaceutical companies didn't use them. But needless to say, it's certainly proved the theory. And now, with CAR T for sickle cell, we have the first use of CAR T for essentially gene therapy. And although it's rather awkward and difficult to apply, it does work. It has been approved. And we're on the verge of a whole new world. So it's a very, very exciting time where different fields of technology have come together. New technologies are emerging. And we're just at the beginning of that revolution. Those of you who get my newsletter, I'll sort of keep you apprised as to what's happening in this exciting field. But it definitely is a major advance in our ability to modify living systems, animals, plants, and ourselves for the better. Will we permanently change the germline of human beings? Probably not for a very long time, if ever, especially as we have treatments that can quickly remedy whatever genetic defect is there. There'll be no need, if we can do it quickly and cheaply, there'll be no need to change the germline. An inherited germline, we can fix up many, many things. There's another revolution that I'm sure that many of you are paying attention to, and that is DNA technology is becoming so rapid that when a baby is born, within 5 to 6 hours, you can have a complete genome read, analyzed, and acted upon medically. That is happening in certain very limited areas today, but will be, I think, common practice, just as putting iodine in the eyes and helping kids with blindness, with doing other more crude tests to look for genetically inherited diseases. People will be doing complete DNA sequencing of newborns, looking for inherited diseases that may have come from the parents, that may have been spontaneous to that child, and they're beginning to apply the therapies right away, shortly after birth, within hours of birth, applying the right kind of therapies to make sure that there'll be a healthy child. That is a reality today in very limited circumstances, but I foresee a time where that will be standard treatment. That plus then they may get a shot to edit the genes, a CRISPR shot to edit the genes, and you'll have a healthy child, where otherwise you might have decades or more of suffering. So this is truly an exciting time. It isn't one without downsides. It's not one that doesn't have more refinements needed, but it is a first step forward on a very long path to improve health for everyone. So I want to thank you especially those in the US, for getting up early. I want to thank you for those who are abroad for staying up. And I hope this has been useful for you. Take care.